Assalamu alaikum. Welcome to Wetlek. Today I am going to discuss the largest gland of body, which is liver. So all the substances that are transported to gastrointestinal tract after being digested are passed to the liver to be by being absorbed in the blood, and they are passed to the liver so that the toxins and drugs and other such harmful substances may be detoxified in the liver. So the blood that transports the dissolved nutrients to the liver actually transports this blood from GIT via portal vein to the liver. So in this way, liver receives the blood which is rich in nutrients and other hormones through portal vein. Splenic circulation is the blood flow to the abdominal gastrointestinal organs, which include stomach, liver, spleen, pancreas, small intestine, and large intestine. And in this planktonic circulation, a significant amount of oxygen has been utilized. So the blood which is going to the liver through the portal vein is not very rich in oxygen, even though it may be rich in nutrients, hormones, and other substances. But it is not rich in oxygen. So it is, we can say that the blood that is going via portal vein to the liver is deoxygenated blood, because its oxygen content has been utilized by the splenic circulation. Liver also needs oxygenated blood, which is supplied by hepatic artery. So the hepatic artery delivers oxygenated blood from the general circulation to the liver. So up till now we have discussed that liver receives blood supply from two sources. The first source is the hepatic artery, which delivers oxygenated blood from the general circulation, and the second is the hepatic portal vein, which delivers deoxygenated blood from the small intestine containing nutrients. This hepatic artery divides into arterioles, and these arterioles reach the hepatic lobules of the liver, and at each corner of the hepatic lobule an arteriole is present, which is bringing the oxygenated blood to the hepatic lobule. And there is also a branch of portal vein on the at the corner of the hepatic lobule, which is bringing the blood, which is bringing the nutrient rich blood to the hepatic lobule. Hepatic lobule is actually an anatomic unit of the liver. These hepatic or liver lobules are polygonal or hexagonal, and are composed of plates of hepatocytes which are radiating outwards from the central vein to the portal triads. So at the center of each hepatic lobule, each hexagonal hepatic lobule, there is a vein which is called central vein. Now, from the corners of the lobule, hepatic lobule, sinusoids reach at the center of the lobule towards the central vein. And in this way, both hepatic artery and portal vein drain their blood into the sinusoids. So we can say that this blood flow is centripetal blood flow, because it is flowing from periphery to the center of the lobule towards the central vein. And one important thing is that the blood that is flowing in the sinusoids is no mixed blood because it has been mixed, because the one of the blood was the blood that was coming from hepatic artery was fully oxygenated blood, while the blood that was coming from portal vein was deoxygenated blood and rich in nutrients. So when these both bloods mixed in the sinusoids, we can say that sinusoidal blood is a mixed blood. As I have said previously, that hepatic lobules also contain the hepatocytes, which are radiating from the central vein to the portal triads. So these hepatocytes are composed of plates or one cell thick layers that are bathed on their either side from the hepatic sinusoids. So hepatocytes are bathed on either side by blood from the hepatic sinusoids. Between each row of cells in a small space is created by caviations in the plasma membrane of two opposing hepatocyte cells. So between the plasma membranes of the two hepatocytes, there is a cavity or small space. This plasma membrane of the hepatocytes, which is lining this cavity or this small space, is isolated from the remainder of the plasma membrane by tight junctions. Let's repeat it. Between the plasma membranes of two hepatocytes, there is a cavity or small space, and this cavity is lined by the plasma membrane of both hepatocytes which are forming this cavity. And the plasma membrane of these two hepatocytes is isolated from remainder of the plasma membrane of hepatocytes by tight junctions. So within the plates of hepatocytes, these caviations or these small spaces join to form channels or canaliculi, and these canaliculi then connect to the bile ductules. So bile is secreted from hepatocytes, and from hepatocytes it goes into the canaliculi, which is formed between two hepatocytes in the plate of cells of hepatocytes. So bile is secreted from hepatocytes into canaliculi, from which it then flows into the bile duct system. So flow of the bile is centrifugal, as it is flowing from the center of the lobule to the periphery. From a functional standpoint, we can also say that canaliculi is an acini which is lined by hepatocytes and is emptying into the biliary duct system. And this biliary duct system is drained by bile which is secreted from hepatocytes. The epithelium of this bile duct is metabolically active, and it is capable of altering the composition of canalicular bile by adding additional water and electrolytes, and one of the electrolytes is bicarbonate. So the composition of canalicular bile is altered by the epithelium of bile duct. There is one other structure in the hepatic lobule, which is a space of DC, and this space of DC is we can say that it is a subsinusoidal area, or it is an area which is just outer to the sinusoids. So between the hepatic sinusoid and hepatocytes, space of DC is present, and through this space of DC, there occurs the exchange of substances between hepatocytes and sinusoids. So the space of DC, which is a subsinusoidal area, or which is present between hepatic sinusoids and hepatocytes, functions in the exchange of substances between the hepatocytes and sinusoids. So we can say that hepatocyte has one phase to treat the blood and other phase to treat the bile. So the phase which is facing the sinusoid, or which is adjacent to the space of DC, is facing the blood, and other phase is treating the bile. Liver sinusoids are actually porous, and plasma leaks from liver sinusoids into the space of DC. But most of the plasma which is leaking the liver sinusoids and is coming into the space of DC also drains back through these holes into the liver sinusoids. But some of the plasma doesn't drain back into the liver sinusoid, and this plasma will be converted into lymph, and this lymph will also flow outward, or its flow will be centrifugal. So we can say that the corner of the hepatic lobule actually accommodates four systems. That is, it has a branch of portal vein, it has a branch of hepatic artery, it has a lymphatic drainage system, and it has a bile duct drainage system. Central vein takes the blood from center of the hepatic lobule and drains into the sublobular veins, and these sublobular veins join to form hepatic vein, and hepatic vein drains into the inferior vena cava finally. And the blood which is drained from hepatic vein into the inferior vena cava is deoxygenated blood, is purely deoxygenated blood. As bile is secreted from the canaliculi to the bile duct system, these bile ducts branch from each corner of the hepatic lobule, from the canaliculi of the hepatocytes, to form a right and left hepatic duct, and these both right and left hepatic duct of the hepatic lobule combine to form a common hepatic duct. This common hepatic duct then combines with the cystic duct which is coming from the gallbladder, and then it drains into a common bile duct. So the cystic duct from gallbladder and common hepatic duct from the liver combine to form common bile duct. Then this common bile duct fuses with the pancreatic duct from the pancreas, and they both combine to form hepatopancreatic duct, which opens into the duodenum through ampulla of Vater. In this way, bile that is formed in the liver hepatocytes but stored in the gallbladder is transported to the duodenum along with the pancreatic juice to undergo further digestive process. Now let's talk about some functions of the liver. Liver is exocrine part of the exocrine part of the liver secretes bile. The endocrine part of the liver secretes somatomedins, angiotensinogen, thrombopoietin, and hepcidin. One of the major functions of the liver is the breakdown of old erythrocytes. Buffer cells which are present in the liver detoxify toxins. Liver also causes the synthesis of various nutrients and metabolites like glucose, proteins, and lipids. Liver also forms urea, uric acid, and it also stores glycogen and iron. And liver also collects nutrients from the small gut, which I have discussed already. Now let's talk about bile. As I have discussed previously, that bile is formed in hepatocytes, and it is liver hepatocytes, and it is then secreted to the gallbladder for its storage. Bile is the actually the excretion of liver, and it is essential for fat digestion or emulsification of fats, and along with various enzymes. The composition of bile is such that it contains 97.4% water, while 2.6% solids. Out of this 2.6% solids, there are organic constituents and inorganic constituents present in the bile. The organic constituents of bile form 75% and include bile salts, bile pigments, mucin, cholesterol, neutral fats, fatty acids, phospholipids, and alkaline phosphatase. While inorganic constituents include sodium, potassium, calcium, magnesium, calcium palmitate, and bicarbonate ions. Bile is actually alkaline in nature with pH of 7.8 to 8.6, and this alkaline pH is because of the high levels of bicarbonate ions that are present in this bile. Now let's talk about bile salts. Bile acid like cholic acid occurs mostly in combination with sodium and then forms bile salts, and the function of bile is due to the presence of these bile salts. Liver then conjugates these bile salts with glycine and taurine and forms glycocholic acid, taurocholic acid, deoxycholic acid, and taurochinodeoxycholic acid. These salts are reabsorbed from the small intestine by enterohepatic circulation, and they are absorbed 85 to 90% from the small intestine. These bile salts are formed in liver. So these bile salts are formed in liver by the combination of sodium with cholic acid to form bile salts. Now let's talk about bile pigments. Breakdown of hemoglobin occurs in liver, and the breakdown products of hemoglobin. One of the products of hemoglobin breakdown is biliverdin and bilirubin, which occurs in combination with glucuronic acid. So these biliverdin and bilirubin substances are conjugated with glucuronic acid. Now, the presence of any type of infection in the gallbladder produces an enzyme which is called glucuronidase. And this glucuronidase enzyme causes the detachment or separation of the glucuronic acid from biliverdin and bilirubin. When biliverdin or bilirubin becomes unconjugated, then bilirubin combines with calcium and in this way it becomes insoluble in water. And when it becomes insoluble in water, it continues to form stones in the gallbladder and bile duct. Now let's come to the cholesterol. Cholesterol is synthesized by the liver, and it occurs in correct proportion with bile salts. And this proportion is very important, and if the level of cholesterol increases beyond this proportion, then it results in the precipitation of cholesterol in the gallbladder. And when it precipitates, it causes stone formation in the gallbladder. So cholesterol is secreted in the liver. It is synthesized in the liver, secreted in the bile, and reabsorbed in the micelles. A micelle is actually an aggregate of surfactant phospholipid molecule of surfactant phospholipid molecules dispersed in a liquid and forming a colloidal suspension. So a micelle is a colloidal suspension of aggregate of surfactant phospholipid molecules, and these micelles help the body to absorb lipids and fat soluble vitamins. So it causes the absorption of cholesterol also. I have said that gallbladder stores bile. So, what is the need that the bile is synthesized in liver and it is not directly supplied to the duodenum while it is first stored in the liver in the gallbladder, then it is supplied from the gallbladder to the intestine. So, what is the reason? The reason is that the gallbladder concentrates the bile by absorbing its water and electrolytes. So gallbladder mainly absorbs the bicarbonate from the bile and renders it either neutral or acidic with pH of 6.5. Initially, the bile was highly alkaline with the pH of around 7.8 to 8.6 due to presence of high levels of bicarbonate ions. But in the gallbladder, as these bicarbonate ions are absorbed by the gallbladder, it becomes either neutral or acidic, and its pH changes to 6.5. This acidity has an important role, and that role is that this acidity inhibits the precipitation of calcium carbonate within the gallbladder, and in this way it prevents the stone formation in the gallbladder. Gallbladder also adds some mucus in the bile to make it viscous, and in this way it concentrates the bile. Gallbladder also causes secretion of toxins, drugs, copper, mercury. In this way, it causes their excretion from the body because bile is finally going to the small intestine to be excreted from the body. One of the effective method to secrete toxins, drugs, copper, and mercury from the liver is to drain them into the bile, and then gallbladder finally passes this bile to the duodenum, and in this way these toxins are finally excreted from the body. Now let's talk about the functions of bile. First very important function of bile is the emulsification of fats. So what is this emulsification? This emulsification is simply a process of breaking down of the fat into smaller units. It is only the simple breaking, not digestion of food. Keep it in mind. This digestion is only caused by the enzymes, by lipases. Emulsification of fats, which is done by the bile, is only the breakdown of fats into smaller units. In this way, emulsification of fats or breaking down of fats causes increase in the surface area of fats in the small intestine, and by grouping them into the small clusters, and when the surface area of fats is increased by being broken down into smaller cells, then it will be easy for enzymes to function on these fats and to digest them. In this way, fat emulsification initially helps the enzymes to digest fats into fatty acids and glycerol, and then these fatty acids and glycerols are easily absorbed by the small intestine. So the bile salts only break down the fats into small globules so that the surface area of the fats is increased within the small intestine, and when this surface area will be increased, the action of lipases on this high surface area fats will be much effective, and lipases will then much effectively act on these fats and will digest them, and then these digested or these fatty acids and glycerols are easily absorbed by the small intestine. These bile salts also activate lipase. So lipases are secreted from pancreas into the intestine for the digestion of fat, and similarly bile salts are also released by the liver in the small intestine, and these bile salts then activate these lipases for fat digestion. These bile salts also have a strong choleretic effect. Choleretic action means that any agent that can stimulate the liver to increase the output of bile causes the choleretic action. And in this way, bile salts act as a choleretic agent and increase in the bile flow. Bile also functions in intestinal peristalsis because bile breaks down fats into fatty acids which can be taken into the body by the digestive tract. The bile acids actually inhibit gastric emptying and reduce small intestinal transit time. On the other hand, they stimulate colonic peristalsis and increase colonic transit time. So, by inhibiting the gastric emptying, bile salts allow the small intestine to undergo further digestion and absorption of the food the transit time of the food is reduced in the small intestine by these bile acids. And by stimulating the colonic peristalsis, bile acids cause an increase in the peristalsis. The bile also has mild antiseptic effect, and in this way it kills bacteria in the present in the small intestine. Bile salts are also responsible for vitamin absorption. Bile salts help the body to absorb fat soluble vitamins like vitamin A, D, vitamin E, vitamin K. So in this way it causes the absorption of vitamins. Bile pigments also add color to the feces. Bile salts also make insoluble substances water soluble. Actually, bile salts act as emulsifier because they have a hydrophilic head that is attracted to water molecule and a hydrophobic tail that is attached to lipid molecule. So bile salts are able to form micelles, and they can break down large lipid droplets into smaller ones, and increasing the surface area for digestive enzymes. And in this way, we can say that bile salts make water insoluble substances water soluble. Bile salts form complexes or micelles, which I have discussed previously. Bile salts also increase the intestinal absorption of cholesterol. Actually, bile salts form micelles, which consist of a lipid bilayer of phospholipids, and into this lipid bilayer membrane, cholesterol molecule gets inserted. And because of this insertion of cholesterol into the lipid bilayer membrane, cholesterol is finally absorbed from the micelles into the intestinal wall through a protein channel. So in this way, cholesterol is absorbed through the intestine by the action of bile salts. Now let's discuss the regulation of the secretion of bile. Vagus nerve stimulation causes increased bile secretion, and similarly bile secretion is also increased by the secretion of cholecystokinin, that is CCK-PZ, hepatocrinin, secretin, and gastrin from duodenal mucosa when lipid-rich diet is ingested. So when lipid-rich diet is ingested, all these factors cause the stimulation of the release of bile. Now let's talk about the effects of deficiency of bile on the body. So the deficiency of bile causes decalcification of bones. It causes gray colored feces due to the absence of bilirubin, because when bile is deficient, then bilirubin, which is it's one of the pigment, will also be deficient. And we have studied that one of the functions of bile is to add color in the feces. So when bilirubin will be deficient, then gray colored feces will be produced. As bile causes the absorption of fats, cholesterol and vitamins from the body, so the deficiency of bile will affect the fat, cholesterol and vitamin absorption, and they will not be absorbed from the body, and the presence of these fats will be identified in the feces, and this will produce a condition which is known as steatorrhea.